Thank you. I was asked to comment on the role of reproducibility and how far is it a problem just for scientists or for society. And of course, the answer is going to be that it's important for both, but I hope to give some examples to explain why. Before I do that, if I could have the next slide, I would just like to briefly say how nice it is to be presenting to people in Leiden, because I discovered rather recently that Oxford and Leiden do have a link. We are linked cities. We're one of the longest twin linked cities that Oxford has. And there is this nice little plaque that I found when I was wandering around Oxford in one of our shopping centres commemorating it. So very nice to be here. If I could have the next slide. So what is the problem with reproducibility? Well, there's really a number of issues here, which have been around, as I shall show you, for a very long time. But I think a paper that really sort of caught attention, largely because of its title, was this paper by Johnny Anides that he published back in 2005, saying why most published research findings are false. And he sometimes lambasted for overdoing things and for exaggerating. And here he didn't actually demonstrate that they were all false, but he showed a lot of reasons why one would not have confidence in research findings. And around the same time, there was growing concern, interestingly enough, from pharmaceutical companies. And there was a little piece published when reporting that Bayer had tried to build their drug development on a number of studies from university research, which had actually not replicated. So they were hoping to develop and take things further, but they couldn't get past the first result in a high proportion of cases. And this seemed rather shocking, given that these were papers coming out of university departments that seemed to be publishable and give strong results. And there's a quote here from Bustin, who discussed this concern about the reliability of particularly biomedical research. And the fact that this leads to a lot of waste. If people are coming up with stuff, getting it published, but then other people try and build on it, you can really waste a lot of time and money. This was a point also raised in this Lancet article. There was a little symposium held, particularly on the topic of research waste, which made the point that you get, on the one hand, sometimes people doing things they don't need to do because we already know the answer, but also people doing things that just are not solid enough. But it was Brian Nozick who I won't say much about Brian's work because you're going to be hearing from him, but I think this was the first really large-scale empirical demonstration of the problem in psychology. So they were taking papers from 3 quite reputable psychology journals and literally trying to replicate them by recruiting squads of people to redo the studies, often with larger sample sizes than they had initially. And really, it depended exactly how you defined replication, but there were really typically 30–40% of things that were replicable, but a lot of them not replicating or not even coming close to doing so. Could I have the next slide, please? And so I talked about, when I try and explain why is this the case, I have talked indeed about the 4 horsemen of the apocalypse. And if we could have the next, one is p-hacking, and the next publication bias, the next low power, and the next harking, hypothesizing after results are known. And I'll just put each of these in their historical context. So if we have the next slide, we're going to start with p-hacking. Now, you will be aware, I'm sure, of De Groot, who was a very famous chess player, apart from anything else, but also an academic, who back in 1956 wrote a brilliant piece about the problems that we get if scientists mix up hypothesis testing and hypothesis generating research and then apply statistical tests. And the point that is made here is that we need, of course, in science to do exploratory research. That's how we get our hypotheses. We observe phenomena, we look for patterns in data, and then we see that there is indeed a hypothesis that we can test. The problem is if you try and use the same data to both generate the hypothesis and then to test it. That simply doesn't work because the statistics are all skewed, because you've basically capitalized on chance. When you have formulated your hypothesis that way, you'll be seeing some patterns that are not meaningful. But then if you start saying, well, that was my hypothesis, it really messes things up. And he explained this beautifully back in 1956, but he wrote in Dutch. And so not much notice was taken of it. But it was recently translated by E.J. Wagenmakers and colleagues. And this piece I always recommend to people as a very good account of what the problem is. Next slide. I'll illustrate it in one way, but the idea is there's many, and this is the point he makes, is there's many different things you can do when you're exploring data, and you often don't know how many there are. There's a sort of like an unconstrained set of possibilities of what you can look at, how you analyze it, and so on. One way is to take a big data set and to analyze it in many different ways. And in the field, people often talk about the garden of forking paths, using this Bourget's short story as a sort of allegory for it, where you can go down all these different paths and where you end up might be quite different. If I go to the next slide, this is my little illustration of it. To suppose that I am interested in handedness, which I am, and whether it relates to developmental disorders. Somebody gives me a very big data set on ADHD and handedness, and I look for an association. And what do I find? I find nothing. I find that there's really not much evidence for that. But I decide to look a little bit more in the data. And if I'm next slide, and what you see then is that we start seeing that in younger children, there's a hint of an effect there, not in the older ones. So I start to think, well, this may just be specific to young children. And then next slide, I go on and think, well, also we've got two measures. We've got a preference measure, which is P, and a skill measure, which is S. And if I look at the skill measure, it's starting to look really promising. Now, what you may have noticed in the bottom left hand of this slide is that I'm putting the probability of a significant so-called p-value in the bottom here for each time we go down this forking paths. And the point is, it's not constant at 5%. Because there's all these different paths you could have taken. If I'm going to say I'm interested in anything, going down any one of these paths, then I need to take into account the fact that I'm really, in effect, looking at many different possibilities and getting excited at any one of those. That's a different probability from the sort of probability you get when you just test for one thing. So we've now got actually a 20% chance that at least one of these things is going to be significant. Let's go a little bit further. Next slide. So now we say, let's look at males and females, and look, the females are looking very, very promising. And we do one more down the forking path, next slide. And we find that if you look specifically on young urban females on a measure of hand skill, P creeps below 0.05, and you rejoice and you think, I have a publishable result, and you invent some story as to why this makes sense. But by this point in time, the chances of you finding something, had you explored all these little forking paths, is over 0.5. So if you look at enough things, something will pop out. And this is the problem, the curse and the problem of p-hacking. Many ways you can p-hack. This is just one example, subdividing into subgroups or taking different measures and from many measures that you've got. Next slide. Other thing is publication bias. Again, we have known about this for years. This goes back to the 1970s at least. Greenwald famously talked about prejudice against the null, although Rosenthal had the much more snappy idea of the file drawer problem, which we can all relate to. You've got a file drawer full of the things you never published because they didn't work out. And Greenwald makes the point that if you actually are prejudiced and you don't like to publish null results, you can end up with a whole load of things that are published that are very misleading because they're not representative really of results in the field if you only see the positives. And as he said, the publication system may be regarded as a device for systematically generating and propagating anecdotal information. Next slide, please. Newcomb talked about low power. Low power has been talked about on and off. In fact, before Newcomb, Jacob Cohen was very famous for highlighting the problem that many, many scientists still don't really get taught about or don't take seriously, which is that we have to worry not just about the false positives from the p-hacking and so on, but also negatives, false negatives that you can get. You may fail to find a result that looks statistically significant even when there's a real effect there, particularly if your sample is rather small. And small samples are the curse of the field because obviously gathering large samples is expensive and it's often difficult if you're working with rare groups or working with animals, for example. It may be difficult to do large-scale studies. And the problem is that what he points out is that people still go ahead even if they may only have, say, a 20% chance of seeing the effect if it is really there. And this is a very wasteful way to proceed, to do low-powered studies. But it's in the field of clinical trials, they're really on top of this and they realize what a problem it is, and you're really typically not allowed to go ahead unless you've got a sensible sample size. There's many, many other disciplines where that's not true. Next slide. And then there's Harking, which I don't know whether outside psychology people are so familiar with this term, but it stands for hypothesizing after the results are known. And this was talked about in 1998 by Norbert Kerr, although again, it goes back to Groot, who very much was saying that this is what people do. They find something, they explore their data, they find something that looks plausible, and then they say, that was my hypothesis, but it's after the results were already known. I recommend this paper very much. It's a very interesting read, and it's written in this wonderfully discursive style that existed back in the 1990s. But next slide shows you why he was concerned. This was the sort of thing that was going on in our field. Daryl Bem, very, very eminent psychologist, was writing this practical guide. In fact, this is the second edition. In the first edition, he said much the same, and that was what prompted, to some extent, the Kerr paper on Harking, because what Bem was encouraging people to do was to do exactly what you're not supposed to do. And so this illustrated really just how normative this kind of behavior was, and often in many areas still is. People think this is what doing science is all about. And it was just a failure to really realize how distorting this is. But if you've got an authoritative person recommending that you should not write the article you planned when you designed your study, but as it says here, you should make the write the article that makes most sense now that you have seen the results. This is really encouraging people to do things backwards. And he's again also recommending p-hacking, look at your data from every angle, analyze the sexes separately, make up a new composite. This is really telling people you've got to p-hack to find something in your data, and then you can tell a story about it. So this is how not to do science, but it has been very much pushed upon people. And I always get particularly upset by the quote at the bottom, which comes from the back of this book, which tells you that the book gives you guidance that will help new academics plan, play, and ultimately win the academic career game, which I find an appalling view of why we're doing science. We are not playing a game. Science should be cumulative. And it goes back to Isaac Newton, who claimed that, he had made progress by standing on the shoulders of giants. Newton was not a very modest man, so it's a rather surprising quote. But the idea is that we progress not just by doing one wonderful experiment, but by building on what's gone before. If I can have the next slide. The problem with the sorts of influences from these four horsemen is that you get early career researchers in particular who have a really tragic situation, because they come along and assume that what they've read in the literature is good, solid stuff that they can build on. And it's a very common experience, and I'm always getting people coming to me and saying this has happened to them. You start on doing a PhD, you think that you'll be able to take a result from the literature and build on it and develop a new idea, only to find that you can't replicate that initial finding. And you think initially it's your fault and you've done something wrong because you're inexperienced. And then other people may say, oh yes, we never managed to get that either. But it's very, very unclear, just when you start out, just how unreliable the literature is. And this is really a serious problem for science, that there's a lot of waste again, and we waste good people who go and leave the field because of this. If I could have the next slide. But then we come to the question of society, which was in the title and I was asked to address. And I would say that the bigger tragedy as well is for society. And this, my goodness, it's very apparent now when we're in a pandemic and we depend on good science. We cannot afford to have rubbish in the literature and non-replicable findings. We can't afford to waste the resources that we've got. Yet this has gone on. And one of the reasons I wanted to introduce it historically was to say, it's not that these problems are new. We've known about these problems for decades, but they still go on. And Buston made that point, that there's been reports about poor quality of research methodology, inadequate implementation of research methods, inappropriate analysis procedures, lack of transparency, and nobody basically has done anything about it. All has failed to stir researchers, funders, regulators, institutions, companies into action. What has changed, and the reason people say, well, does this mean this is going to go on forever? And I don't think so. And I know Brian is going to talk about some solutions, because I thought we decided I'd talk about the problems and he would focus on the solutions. But I think there's also an appetite now for change. But the interesting thing is it's coming not really just from academics, but it's coming from those who use research. So doctors and patients, pharmaceutical companies, who are often painted as the villains because they make money out of drugs, but actually they don't want to waste their funding, and they've been very vocal in pushing for better standards. But the other big thing that's changed is the advent of social media, and these issues are greatly discussed on social media. And again, it comes back to these often these early career researchers who have been burned, had their fingers burned trying to do good science, only to find that all the things that they were hoping to work on are not really very solid. And I think those influences now have combined to mean that we're in a much better position to tackle this. And the next slide. But really, what are we trying to do in science? We're trying to move from a position where we don't know about anything to where we are very solidly aware of what the facts are. And these are referred to sometimes as canonized information, or canonized facts, when something is so certain that it would actually be rather pointless to try and do a study on it because we know the answer. So the Earth is round, well, spherical. Global temperatures are increasing, I think has moved up into that green zone, or in a much more mundane level, caffeine makes you wakeful. If you started trying to publish a paper that said that was not true, you'd have a hard time because people have got a strong prior. If you go down to the bottom here under the red, there are other things where there's overwhelming evidence against them. If you tried to publish a paper saying that astrological forces determine your fate, you would have a hard time in a science journal because people just don't believe it. Extrasensory perception is real is an interesting one, because Darrell Bem, who wrote that famous paper, did try to publish and succeeded in publishing a paper claiming there was some evidence for that. And that was one thing that actually kick-started psychology's concern about reproducibility, because so many people just thought this isn't actually true. But when we do research, we're mainly focusing on things in the middle, and we're trying to move to the green or to the red. And hydroxychloroquine is effective against COVID-19. We realized that there was a time when that was really a live issue, and we were moving a bit up to the green, and then I think we've moved well down into the red now. But you need a series of studies, a series of believable studies with strong evidence. It's not ever a one-off thing. This comes back to the idea of science being cumulative. You have the next slide. This was talked about by Nissen and colleagues, who did a very nice bit of modeling about how false facts get canonized, and showed that you just basically can get accumulation of evidence that looks very strong, but is not strong because of things like p-hacking and publication bias. So you get these influences that are really meaning that instead of having a solid body of evidence that you think you've got these biased sources. Who's to blame when this happens? So I chaired a symposium back in 2015 on this, where we tried to look at this. And of course, there's no one person to blame. There's a number of people who are, not people, but sort of institutions and actors in this scenario who are all involved. And this is a multiply complex problem. So we'll just look at some of the examples. Okay, I could have the next. So I think we need to be aware of the roles played by researchers, by journals and indeed publishers, by institutions and by funders. So let's have a look at some examples, if I can, next one. So with researchers, we have got the problem of the poor grasp of statistics and probability and methods. It's evidence when I say, people are doing p-hacking, harking, without even realizing this is a problem. And that's often down to bad training. But then there are other things like a reluctance to collaborate with competitors, which I think Kurt mentioned in his introduction, a reluctance to share data, even fabricating data. And these are things that also conspire to mean that, fabrication is an extreme example, but these reluctance to collaborate, reluctance to share just makes it harder to check things and harder to detect errors if there are errors in work. And one of the things that many scientists, I think, get upset about is the idea they might have errors in their work. But the truth of the matter is we are all going to have errors in our work. Errors are ubiquitous, and the thing we need to be is less, hung up about that, and more trying to work in a way that will ensure they get detected, assure that they get detected if they occur. I'm going to have the next slide. So yes, solutions. What would the solutions be? Well, one of the things about the last three of these is that one can see that they're going to be heavily influenced by incentive structures. If there's no incentive to collaborate, if it's actually seen as a bad thing, or to share data, just a waste of your time, doesn't do any good, or if there's real pressure, such pressures to produce a sexy result that you fabricate data, this is coming down to incentives. And that's where we move on to these other players in this space. I've been blogging for years about problems caused by journals and publishers. Largely when I get irritated, I blog. Maybe I've had a bad experience with a paper I've written, or I've read something that's irritated me. Over the next slide. So this is, again, reiterating some of the points that Kurt had made. Journals are often, particularly the so-called top journals, are often really hung up on newsworthiness. And sometimes to the extent that it really trumps any concern about methodological integrity. What they should be doing is looking first at what is methodologically rigorous, and then perhaps worrying about if it's newsworthy. But I've used an example here, and I blogged about it, of a paper that never would have got accepted had it not come up with a very sexy result. This is also true with a reluctance to publish so-called negative findings, null results. There was a paper showing that on brain scanning that you could distinguish Christians from atheists or something. Can you imagine anybody publishing that result if they hadn't found a positive result? Of course they wouldn't. It's totally boring. And again, one's Bayesian prior for the likelihood of this being true is extremely low. But if you get somebody doing that, even in a small, not very impressive study, it makes news. And so it does get published. Let's have the next, yes, replications or failures to replicate also often dispreferred by publishers and journals, even though to my mind, until you've replicated something, I often, in many fields, don't believe it very much. You need more than one study, and yet they're seen as unexciting. So we have this real problem of these are all things that contribute to publication bias, factors that journals and publishers regard as not very interesting. Next slide. And next slide. So how common is publication bias? This is a really lovely study done by De Vries and colleagues, where they were able to look at it by looking at outcomes of trial registries when they had studies looking at the impact of intervention for depression. And next slide. The reason they could do this study was because for clinical trials, you have to register your trial in a registry these days. So you can pick up on studies that never saw the light of day, didn't ever get published. And what you can see immediately going from that first bar to the second is that red blobs, which are studies that obtain null results, tend not to get published, whereas green studies, which are the ones which found a positive result, will make it into the journals. So you've got this massive loss of material about null results. Then you have what's called reporting bias, which is the next third bar along, where you have studies which actually report different outcomes from the ones that they had registered in the registry, because they might have said, we're going to measure depression this way, but then they find a result that's only significant if they look at it a different way. So that's also a form of p-hacking. And then, as you can see on the next bar along, there's something called spin, which is that, even if you didn't get the result you expected, you describe it as if you had. But perhaps the most damning one is the fifth column. And I'm starting to think we need a fifth horseman of the apocalypse here, because this is something I really started to get more and more interested in citation bias. And this is very much the fault of the scientists themselves, because this is a tendency to only cite material that agrees with your preconceptions. And what is interesting in this last column is the size of those blobs reflects how commonly the work was cited. And so I feel very sorry for those little red blobs at the top, because here are papers which managed to survive all these other stages. They had a null result, but they actually got published. They didn't get spun, they didn't get distorted. Yet look what happens to them then. Nobody cites them. They're not reckoned to be interesting. So I think this study is a beautiful demonstration of how publication bias and citation bias conspire to really completely change the picture of what's going on, from suggesting that really there's maybe not so secure evidence for some of these treatments for depression to suggesting that they all work wonderfully. In addition, we then have I won't say much about this today, but I think it's worth a mention because, again, it's a growing concern of mine that we have further problems that I think do have to be put at the feet of the journals and the publishers, which is that where you get evidence of studies being published which are just untrue, either because of honest error or more commonly because of fraud, they tend to stick in the literature. They don't get retracted. And this has been becoming a topic of some discussion as to why this is the case. And I think it's very interesting that the pressures and the concerns of publishers are very different from the concerns of researchers. The publishers are concerned very much with making sure the article gets out there, and they're worried about the authors, and they may be worried about being sued by authors if they were to try and retract work that was suggested to be fraudulent. And of course, they're very concerned not to accuse people of fraud. But the authors and the researchers themselves who read the work are much more concerned about the integrity of the scientific record, and that it's very important to take things out, or at least flag them up as problematic if they are found to be problematic, and to do so promptly before they have a big effect on other people's research. And so I think this is an area I'm interested in having more discussion about, because I think it's really two constituencies with rather different views of what publication is. But certainly my concern is to make sure that the publication record is as accurate as it can be, and to perhaps give a better reputation to retraction, and ensure that it doesn't necessarily mean that somebody's done something fraudulent, but that sometimes things go wrong, and if they go wrong, they shouldn't be in the publication record. I could have the next slide. So we have this, I have this idea that what we have actually got is what, instead of having cumulative science, which is I think what we need, is we've got what I call quasi-cumulative science, where you've got this is going back to that Nissen paper, you've got a picture of consistent support for a hypothesis when the reality is much more mixed, or even may go against it. This isn't just a problem though for researchers themselves and for journals. It's also down to some extent to the funders, because I would argue the funders, as well as the journals, tend to be obsessed with novelty. If I could have the next slide. This, I just pulled out these two examples, and indeed it's two examples from funders who have funded me, so I shouldn't be too rude about them. And indeed I have a very positive attitude towards both of them. But it is noteworthy that if you look at the websites of most funders, they are emphasizing the importance of novelty all the time. And indeed, I've sat on grants panels where the way you kill a project is to describe it as incremental. And if you are applying to the ERC, the European Research Council for funds, they want you to say that you've got a groundbreaking high-risk project. And if you apply to the Wellcome Trust, they say, is your proposal just a direct continuation of existing work? As if that's sort of a bad thing to start a line of work and then continue with it. Next slide. So again, at my blog, I go on about things, but I became absolutely stuck on this issue because it seemed to me that when you think about it, if you look at some of our Nobel laureates, for example, they typically are people who do study one particular question in great detail over many years. They don't hop about. So I drew a distinction between the frog on the lily pond, which I think is a model of many researchers these days, encouraged by the funders to publish a paper on something and then hop off onto the next lily pad and look at something else, and then hop off and look at something else. And you never really get any sense of cumulative research or somebody really, shoring up their findings. You get your paper in Science or Nature, and then you go on to the next thing and you do the next high input thing. I think we should be more like termites who build this mound with lots of little termites coming and adding something to it, and really then getting an edifice that has got some sort of structure and solidity to it. And as I say, if you look at the careers of most Nobel laureates, it's exactly that. In the lab, checking something out, trying to get it right. Look at people like John Sulston, working on DNA, and it's just like, this is what they do. They don't hop about. So it seems to me very weird that we've got this concern to always be novel. Sure, we don't want people to be doing tedious things, and just, I think the worry is otherwise we'll just get people stuck in a rut doing things that we already know the answer to. But I think you can avoid that without this insistence on novelty all the time. Next slide. I just want to finish with a quote from Ottilie Laser, who I'm pleased to say is now the chief executive of our funder UKRI, which is the big overarching UK research institute now. She's very good on this, and that's great to have somebody at the head of UK research who feels exactly as I do about this. She feels that the reproducibility crisis is very much linked to an excessive focus on the publication of so-called groundbreaking results in prestigious journals. She's a plant scientist, and again, I think her own research has been very much more in the mold of really trying to get to the bottom of problems by doing a whole series of studies to try and really understand something rather than hopping about always chasing the next new thing. And she says science cannot only be groundbreaking. There's a lot of important digging to do after new discoveries. There's not enough credit in the system for this work, and it may remain unpublished because researchers prioritize their time on the eye-catching papers hurriedly put together. And I think that this is a real problem that we have, and I'm glad to say she's trying to alter the incentive structures to move away from that. She's got this lovely quote somewhere else where she talks about how if you only focus on groundbreaking research, what you'll end up with is a lot of holes in the ground. And of course, that is exactly the problem, that we have all these big dramatic findings, which you can't then build on because they don't actually lead anywhere. The next slide. Yes, and as I hoped, that is my last slide, the next one. So I've got a lot of slides on this general topic of reproducibility, which you're welcome to look at, and which this reference will lead you to. But otherwise, just thank you very much.
Thank you, Dorothy. A few questions, part of myself, but also from the chat. You've described these problems with reproducibility, and I know Brian will talk more about what can you do about it. But when these issues are being discussed, quite often one does say that science has an internal cleansing process. And then one refers to, of course, to peer review. One refers to tenure committees, promotion committees that should, look at these issues and be concerned about them. Because ultimately, it is taxpayers' money that is being used. It's money from funding agencies. Does the internal cleansing of science, doesn't it work then?
I think at the moment it doesn't, and one of the reasons is that what is being recognised, because the one group I said less about are the institutions that do promotions, and both they and some funders, some more than others, have been very obsessed with things like the journal that you publish in, the journal impact factor. And so it is the case still that, I mean, if you apply for a job in a UK university, they'll often say that what they're looking for are two things. They're looking for publications in high impact journals, by which they often mean Science and Nature and Cell, Neuron, a few sort of very, very high impact ones. And they're looking for grant income. And I think it's fine to be looking for grant income in the sense that you want somebody to be able to demonstrate that they can bring in grant income. But the problem is it shouldn't scale with the amount of grant income. Because what you're encouraging people to do is get more and more. And in fact, you get these sort of people who are like captains of industry. They have tons of grants, none of which they can actually run properly because they're too, there's too much to juggle and everything is then delegated down. So I think this focus on having as much grant funding as possible rather than adequate grant funding to do the bit you're doing is one problem. And then evaluating people not by looking at the methodological rigor of their work, but looking at the journals that they published in is a really serious problem. And it's more so in some domains than others. I was interested recently to discover that probably the worst field for this is economics. In economics, there's 5 journals. Apparently, if you don't have a paper in one of those 5 journals, you're not going to make it to ever get appointed as an economist, academic economist in a fancy institution. It's like sort of, you know, the pass that you have to get. So we're trying in Oxford. We've got a Reproducible Research Oxford group set up that the university's helped funding. And we're looking very carefully at, can we change the criteria that people are evaluated on to encourage the sorts of behavior that we think will improve science. But at the moment, if you give the sort of talk I've given to junior people, they will often say, if I do what you say I should do, if I don't PHAC, I don't Hark, and I don't worry about the journal impact factor, then my career is going to be damaged, which is a severe indictment of the current state.
Because it also, of course, begs the question on how young career researchers are being mentored to do good science, while they do see bad examples and they, in fact, encouraged to make shortcuts. Yeah,
absolutely. Again, it's not everyone, but there's always a percentage of people in any audience I speak to who say, well, it's all very well for you to say that, you know, you've made your career, you've done well, but you're telling us to behave in ways that are going to be career suicide. And I think quite a lot of people who have got a strong sense of integrity leave the field if they find themselves working in a research lab or a research institution where they are being encouraged to do that because they know it's wrong. And we don't want to lose those people, of course.
Exactly. You also mentioned that in Oxford, you are addressing that with a reproducibility network. What are the types of actions Oxford or your group has taken in order to change this culture?
Well, it's early days, and Oxford is one of the slowest institutions in the world to ever change anything, but they are certainly concerned that there is a problem. And the nice thing about the group that we have is that it was very much initiated by early career people. It was grassroots that were pushing for this. And then we happened to have that happened to combine very nicely with actual top-down influence from actually government who were getting concerned about this. So the university was on the one hand under pressure from government, also from its early career researchers, and then of course some of its own researchers like myself and many others who were also getting worried about this. But there's a number of initiatives that we're taking, but one is to certainly look at how we evaluate people for promotion and for tenure, and to try and get rid of these things like journal impact factors completely. But funnily enough, when that was first, you know, they've been telling departments about this, the immediate response was, this is how I discovered about the economists, you know, how on earth could we evaluate somebody if we didn't take into account the journal that they published in? And you sort of think, well, read their papers, look at the quality of their work. But this is so foreign to some disciplines and some medical disciplines also. So it's going to be quite a battle to, you know, get this through. But if we can get it into the university's sort of official criteria, then people will be able to complain if it's not done that way. And I think things will change.
Okay. Well, thank you so much. Yeah, there are lots more questions, but we'll take those on during the panel discussion. So Daniel Likens can also use those questions. So thank you so much for your very interesting talk and also challenging talk, of course. Yeah.